Have you ever wondered, how an ordinary, totally harmless bacterium just suddenly transforms into a multi-drug resistant superbug practically overnight?
And we mean literally overnight.
Right, because we always hear about evolution taking millions of years or at least requiring thousands of generations of random mutations to adapt to a threat.
Yeah, that's the standard Darwinian model we're all taught.
Exactly. But in the microbial world, a pathogen can just suddenly wake up with an entirely new set of superpowers,
Yeah.
like the ability to destroy 4 different antibiotics at once. And hint, it is not just a random lucky mutation.
No, it's something much more deliberate. Today, we're going to look at the exact mechanism that makes that rapid, terrifying transformation possible.
It's wild stuff.
It really is. We're going to explore how bacteria physically share their genetic blueprints, the incredibly clever, slightly unorthodox ways scientists first discovered this invisible process, and why it serves as the central engine of the modern antibiotic resistance crisis.
Okay, let's unpack this. Because to understand how superbugs are born, we have to look at this massive stack of microbiological research, lab notes, and structural biology papers you sent us regarding a process called conjugation.
Specifically, we're zooming in on the bacterial mating bridge.
Right, the mating bridge. So our mission for this deep dive is to extract the insights from this research and really figure out how this microscopic machinery actually operates. And right off the bat, looking at these sources, I feel like we need to shatter a really popular misconception because people casually call this bacterial sex, but that terminology completely misrepresents what is physically happening, right?
Oh, absolutely. It creates a fundamentally flawed mental model.
Yeah.
By definition, conjugation is the direct contact-dependent transfer of DNA from a donor cell to a recipient cell. Unlike eukaryotic reproduction, like, animal reproduction, there's no mutual exchange of genetic material to create a brand new, distinct offspring.
So there's no baby bacterium.
Exactly. The total number of bacteria in the room stays exactly the same. No new cell is created.
It's completely asymmetrical then. You have this one-way street where a donor cell targets a recipient and just pushes its genetic information across.
Right, and the mechanics of that rely entirely on a massive, specialized, multi-protein transport apparatus.
Okay.
We call it a type 5e secretion system, or T4SS, along with an appendage called a pilus.
Okay, the pilus.
Yeah, and the donor cell possesses a specific piece of free-floating DNA, a conjugative plasmid that carries the architectural blueprint for building this entire transfer machinery.
And the recipient lacks it.
Precisely.
I was trying to wrap my head around this, and my first thought was that it's kind of like sharing a massive digital file.
Okay, how so?
Well, like, if I want to send you a video, I don't give you my actual phone. I use Wi-Fi to airdrop a copy, and at the end, we both have the video.
Right.
But instead of wireless, these bacteria are building a physical Ethernet cable, plugging it into the other cell, and, like, forcing the download.
That's a great analogy, though I will say airdrop implies a willing receiver who accepts the prompt.
Oh.
Conjugation is much more aggressive. The donor isn't asking permission, it's forcing a connection. But the copying the file aspect of your analogy is spot on. And the molecular mechanics of that duplication, which is a process called rolling circle replication, are just incredibly elegant.
I read about this. It starts with a specific cluster of genes, right?
Yeah, the donor's plasmid contains a cluster of genes called the tray operon. And that operon builds that physical cable, the pilus. Once contact is made and a mating junction forms, an enzyme called a relaxase goes to work.
And from the research, this relaxase acts like a pair of molecular scissors, but it only cuts half the DNA.
That's right. It nicks just one strand of the double-stranded plasmid DNA at a very specific starting coordinate. We call that the origin of transfer, or ORT.
ORT, okay.
So it peels that single strand away and begins threading it straight through the mating channel into the recipient cell. But, if you just push the whole strand over, the donor would lose its plasmid entirely.
Which would totally defeat the purpose of spreading the trait. So how do they both end up with a copy?
Imagine pulling a thread from a spinning spool, but as you pull, fresh thread is instantly woven into the gap you just created.
Oh, wow.
Yeah. As that single strand is spooled out and pushed into the recipient, the donor cell uses the remaining intact circular strand as a template to immediately synthesize a replacement.
And what's the recipient doing during all this?
Simultaneously, the recipient cell takes the incoming single strand and synthesizes its own complementary backing. So by the time the transfer is finished, both cells possess a complete double-stranded, fully functional copy.
That is wild. The donor loses nothing. But it gains a fully converted partner who can now go out, build its own cables, and do the exact same thing to another cell.
Which explains the exponential spread, exactly.
Right. But reading through the metabolic costs of all this in the notes, I
Mm.
mean, building that massive type IV secretion system requires a huge amount of cellular energy.
It's very expensive for the cell, yes.
So why do they do it? Does the donor cell actually get any benefit from spending resources to upgrade its neighbors?
That is a critical evolutionary question. And it's actually one of the most fiercely debated topics in microbiology right now. But hold on to that thought.
Okay, I'm leaving a pin in that.
Because to truly understand the why, we first need to look at how we even know this incredibly complex process is happening at all. I mean, we're talking about microscopic single-celled organisms. In the 1940s, the absolute dogma was that bacteria were clonal asexual organisms that just simply divided in half.
Right, they were basically just tiny little cloning factories. You couldn't just put them under a standard light microscope and watch them build tiny Ethernet cables.
Yeah.
So how did scientists figure out they were swapping genes?
It required a truly brilliant experimental design by Joshua Lederberg and Edward Tatum at Yale back in 1946, work that actually eventually earned Lederberg a Nobel Prize. Yeah. They utilized two different heavily mutated strains of E. coli K12. These were auxotrophs, meaning they had genetic defects that prevented them from synthesizing specific essential nutrients.
I looked at their methodology for this, and they basically put the bacteria on a strict starvation diet.
Exactly.
They used a minimal medium, just an agar plate that contained only raw base salts and sugar. No vitamins, no amino acids.
Right, so if you take strain A, which cannot manufacture the amino acid methionine or the vitamin biotin, and you place it on that minimal medium, it starves and dies.
Makes sense.
And the same goes for strain B, which cannot synthesize threonine, leucine, or thiamine. Individually, neither strain has the complete genetic code required to survive on that plate.
But, and this is the crazy part, when Lederberg and Tatum mixed strain A and strain B together in the same tube, let them hang out for a bit, and then plated that mixture on the minimal medium, a few colonies actually grew.
Which was biologically impossible under the existing dogma. The only way those specific bacteria could manufacture all those missing nutrients and survive was if they had somehow combined their genetic information. Strain A had to have acquired the working blueprints it was missing from strain B, or vice versa. It provided the first definitive proof that bacteria were exchanging genetic material.
But Lederberg initially assumed they were fusing together and mixing their entire chromosomes, right? Like acting like eukaryotic cells.
He did, yeah. It took William Hayes in London in 1952 to correct the record. Hayes realized you always had one specific strain acting as the donor, which he called F positive, and another acting as the recipient, the F negative.
So it wasn't an equal merger.
No, Hayes shifted the entire paradigm. He proved conjugation was not a symmetrical sexual process. If a donor lost its F factor, it couldn't donate anymore. But if you bumped it against a healthy F positive strain, it rapidly became a donor again.
Oh, so it moved through the population like an infection.
Exactly. Hayes reframed it as a directional infection driven by a mobile genetic element, which we now know as the F plasmid.
Okay, here's where it gets really interesting. Because discovering that bacteria swap tiny plasmids is one thing. But then researchers figured out how to use this transfer mechanism to map the entire bacterial genome.
Yes.
And they did it using a kitchen appliance.
They did. The famous interrupted mating experiment of the mid-1950s by Francois Jacob and Elie Woolman at the Pasteur Institute. They were working with a very peculiar variant called an HFR strain. That stands for high-frequency recombination.
Because normally the F plasmid is just floating freely in the cell cytoplasm, right? But in an HFR strain, the plasmid physically splices itself directly into the host bacterium's main chromosome.
Right, and that single fusion changes everything.
Yeah.
Because when that integrated plasmid decides to initiate conjugation, the Valaxis enzyme makes its cut and starts threading the DNA through the pilus just like normal.
But now it's attached to everything else.
Exactly. Because the plasmid is physically tethered to the main bacterial chromosome, it starts dragging the entire millions of base pairs long genome across the bridge into the recipient.
It's kind of like trying to email a single PDF, but accidentally attaching your entire hard drive.
A rather large hard drive, yes. The bacterial chromosome is massive, and that tiny protein pilus connecting the two cells is incredibly fragile.
I would imagine.
Yeah, so in a natural, turbulent fluid environment, the mating bridge almost always snaps before the full chromosome can make it across. Jacob and Woolman realized they could intentionally exploit that fragility to map the genes.
I just love the visual of this. They literally used a Waring blender. They took the donor and recipient bacteria, mixed them together so they'd start mating, and then very specific time intervals, like every couple of minutes, they pulled the sample and threw it into the blender.
The sheer physical force of the spinning blades ripped the microscopic mating bridges apart. It instantly stopped the DNA transfer at an exact known second, while leaving the tiny bacterial cells completely unharmed.
It's the equivalent of pulling the Ethernet cable out of the wall at precisely 5 minutes, then 10 minutes, then 15 minutes.
And then they just analyzed the recipient cells to see which genes had successfully downloaded before the connection dropped.
That is so smart.
By tracking which traits appeared in the recipients at which time intervals, they proved that genes enter in a strict, predictable, linear order.
Right, so if the gene for processing lactose always showed up at the 18-minute mark, and the gene for synthesizing galactose showed up at the 25-minute mark.
They knew exactly how far apart they were. They created the very first genetic map of E. coli, measuring distances not in nanometers or base pairs, but in time, a 100-minute chromosomal clock.
And geneticists still reference the E. coli map in minutes today.
Yeah.
It's brilliant. But, thinking about that blender experiment brings up a major logical gap for me.
What's that?
Well, if this bridge is so fragile that a kitchen smoothie maker can rip it apart, how did it survive natural turbulence in the wild? I mean, in textbook diagrams, the pilus always looks like a rigid, hollow tube, like two bacteria sharing a milkshake through a tiny drinking straw, with the DNA flowing through the middle.
Yeah. That drinking straw image is a massive oversimplification that structural biologists have been trying to correct for years.
Really? So it's not a straw?
No. Recent advances in cryo-electron microscopy, particularly from Gabriel Waxman's lab, have completely challenged that static picture.
So it isn't a rigid pipe bridging the gap?
Not at all. It functions much more like a molecular grappling hook.
A grappling hook?
Yeah. The evidence suggests the pilus shoots out, makes contact with the recipient's outer membrane, and then actively depolymerizes, meaning it retracts. It physically winches the two cells together through the fluid until their outer membranes are practically touching. That minimizes the distance the DNA has to travel.
Wow.
And the DNA doesn't seem to travel through the long external tube of the pilus itself. Once the cells are pulled flush against each other, a separate complex channel opens up directly between their cell envelopes.
Okay, so the pilus is just the harpoon to reel them in, and the actual transfer happens point blank.
Exactly.
And what's wild is that this harpoon system isn't even universal. When reading the sources on gram-positive bacteria, like Enterococcus faecalis, they don't use this grappling hook at all.
They face a very different structural challenge. Gram-positive bacteria have incredibly thick peptidoglycan cell walls. Enterococcus faecalis relies on chemical signaling instead of harpoons.
Chemical signaling.
Yeah. The recipient cell constantly releases peptide sex pheromones into the environment. When a donor cell detects these pheromones, it triggers the production of surface adhesins, which are essentially sticky, clumping proteins.
Oh, so they just bump into each other.
Right. They bump into each other in the environment, stick together like molecular Velcro, and then construct a channel through their thick walls to transfer the plasmid.
The diversity of the machinery is just incredible. But the part of the research that truly broke my brain is that this machinery doesn't even respect the boundaries of the microbial world. Like we assume conjugation is strictly bacteria talking to bacteria. But then the sources introduce Agrobacterium tumefaciens.
The ultimate boundary breaker. Agrobacterium is an agricultural pathogen. It possesses a specialized conjugative plasmid called the T plasmid, the tumor-inducing plasmid. But it doesn't just use this system to conjugate with its bacterial neighbors.
No, it goes much further.
It utilizes its type 5e secretion system to inject a specific segment of its DNA, the tDNA, directly across the rigid cell wall and into the nuclei of plant cells.
It is literally hacking a completely different kingdom of life. It forces the plant's own cellular machinery to read the bacterial DNA, which causes the plant to grow massive crown gall tumors.
Yes, and those tumors act as little specialized factories, produce rare nutrients called opines that only the agrobacterium can eat.
It builds its own grocery store. And what's even crazier is that human scientists stole this exact mechanism. Modern plant genetic engineering entirely relies on hijacking this bacterial T4SS machinery to deliver modified genes into crops.
It does. We simply swap out the tumor-inducing genes for agricultural traits we want, and we let the bacterium act as a delivery syringe. What's fascinating here is how this completely destabilizes our traditional definitions.
How so?
Well, if a bacterium can inject DNA into a plant leaf, or a yeast cell, or even human cells in a laboratory setting, what actually defines conjugation? Is it the fact that two bacteria are involved, or is it defined entirely by the mechanical hardware, the T4SS machinery?
I'm pushing back on the textbook definition here. If I use a USB drive to move a file from a PC to a Mac, it's still a USB transfer. The machinery is what matters, not the operating system of the receiver. If the T4SS is doing the pushing, it should be conjugation.
You're actually aligning with a genuinely contested debate among modern microbiologists.
Mm.
The rigid definitions of the 20th century are really shifting toward mechanical descriptions. But this ability of plasmids to seamlessly jump not just between bacterial strains, but across vast evolutionary distances, well, it brings us to the terrifying real-world consequences of all this.
So what does this all mean? We started this deep dive talking about superbugs.
Right. The most consequential exploitation of conjugation is the global antibiotic resistance emergency. Let's look at a prime historical example, Japan in the late 1950s. Clinicians were suddenly dealing with severe outbreaks of bacillary dysentery caused by the pathogen Shigella. And to their horror, they started isolating Shigella strains that were entirely resistant to 4 different major antibiotics: sulfonamides, streptomycin, chloramphenicol, and tetracycline.
Simultaneously. Wait, if a patient is taking 4 different drugs, the pathogen should be completely wiped out. I mean, statistically, the odds of a single bacterium naturally mutating the exact genetic sequences to defeat 4 distinct chemical attacks at the same time must be essentially zero. Where on earth did it pick up the resistance so fast?
Consider the math. If the mutation rate for one drug is 1 in a billion, the odds of 4 simultaneous mutations are astronomically impossible.
Right.
The Shigella didn't invent the resistance, it downloaded it. Researchers traced the culprit to an R factor, which is a massive conjugative resistance plasmid called R100.
Did it get it from something else in the gut?
Precisely. It was jumping back and forth between the invasive Shigella and the completely distinct, naturally occurring E. coli bacteria that live peacefully in the human digestive tract.
Oh, wow.
The gut microbiome was basically acting as a silent reservoir. The harmless bacteria were carrying the resistance plasmid, and as soon as the Shigella arrived, they formed mating bridges and just handed over the blueprints for survival.
That is terrifying.
It was the earliest, clearest clinical warning that conjugation, not random mutation, was the engine driving the rapid evolution of multi-drug-resistant pathogens.
And we completely ignored the warning. Because today, we cram dense bacterial populations together in hospitals, and we pump massive amounts of antibiotics into livestock operations. We've essentially created the perfect evolutionary pressure cookers.
We really have.
Just look at the sources on NDM1. It's a resistance gene discovered in 2008 that produces an enzyme capable of destroying our absolute last resort antibiotics. And because it sits on a highly mobile conjugative plasmid, it spread to every populated continent on Earth in just a few years, just jumping from species to species via these mating bridges.
It converts a routine, easily treatable infection into a lethal, untreatable one that is capable of sweeping through an entire intensive care unit in a matter of days. Which brings us all the way back to the question you asked earlier.
Right. Why does the donor cell do this?
Yeah.
Why expend the massive metabolic energy required to build a type 5 secretion system just to upgrade a neighbor?
Exactly. Because Laderberg and the early pioneers assumed it was a cooperative adaptation. They thought the bacteria were working together to increase their collective genetic diversity to ensure the survival of the species.
That makes sense intuitively, right? Like teamwork.
It was a very romantic traditional view. But evolutionary biologists crunching the data look at these plasmids and note they often impose a heavy fitness cost on the host bacterium.
Right, because manufacturing the dozens of massive proteins required for the pilus and the secretion channel exhausts the cell's resources.
Exactly. A bacterium carrying a large conjugative plasmid frequently grows much slower and divides less often than a bacterium without one.
Well, if it slows them down and drains their energy, it's not a cooperative survival strategy. It's a parasite.
This is known as the selfish DNA hypothesis. The plasmid isn't trying to help the bacterium. The plasmid is only trying to propagate itself. It behaves exactly like an infectious agent. It persists in the population, not because it's necessarily beneficial in all environments, but because its rate of horizontal transfer outpaces the metabolic cost it imposes on the host.
So the bacteria are the victims. They're just vessels being hijacked by the plasmid's relentless need to replicate and spread.
If we connect this to the bigger picture, bacteria are not just passive victims of this selfish DNA, they actively fight back.
How?
When we talk about CRISPR-Cas systems, which the public knows as a gene editing tool, we are actually talking about a bacterial immune system. When a plasmid tries to conjugate into a cell, the bacterium uses CRISPR to archive a tiny mugshot of the invading DNA.
Oh, I see.
Yeah, and if it recognizes that sequence again, it deploys Cas proteins like molecular scissors to literally chop the incoming plasmid DNA into useless fragments before it can establish itself.
So conjugation and bacterial immunity are locked in this endless microscopic arms race.
Pretty much, yeah.
The plasmid builds a grappling hook to force its way in, and the bacterium uses CRISPR to try and slice the cable and destroy the incoming code.
It is a constant, brutal war playing out at the nanoscale, determining the evolutionary fate of the microbial world.
This is just mind-blowing. We started with the idea of fake bacterial sex. We looked at scientists sacrificing kitchen blenders to map genomes by the minute.
The wearing blender, yes.
We discovered that the textbook drinking straw is actually a retracting molecular harpoon, and realized this exact mechanism is the engine driving the global superbug crisis. And for you listening, just think about this right now. Inside your own gut microbiome, there are trillions of bacteria.
Countless numbers of them.
Countless numbers of them are firing these microscopic harpoons at this very second, swapping genetic traits, fighting off selfish plasmids, and actively shaping your metabolism and health.
It is happening constantly in every environment on Earth. And I want to leave you with one final thought based on the absolute cutting edge of the research you provided.
Okay, lay it on us.
We know the bacterial mating bridge is the weapon that spread antibiotic resistance like wildfire. But scientists in synthetic biology and phage therapy are now engineering their own conjugative systems.
Wait.
They are designing modified plasmids carrying CRISPR-based anti-resistance constructs and attempting to deliver them directly into pathogen populations.
Wait, they are hijacking the harpoon?
Yes. If conjugation created the superbug overnight, could weaponizing that very same mating bridge to force pathogens to download their own destruction be the ultimate way to reverse the crisis?
Now that is a thought to chew on.